Hi friends, my name is Preeti. So today I'm going to discuss about the remaining neurotransmitters. So the first one is GABA, which is GABA aminobutyric acid, and it is an inhibitory neurotransmitter. Okay. So first we'll see the types of GABA receptors. Okay, there are two types of GABA receptors, GABA A and GABA B. GABA A is a ligand-gated ionotropic receptor and GABA B is a GPCR coupled, G protein coupled. Okay. So GABA, when acts on GABA E, it results in influx of chloride ions and there is a hyperpolarization which leads to inhibitory neurotransmission and there is no excitation. And if we come to GABA B, it is a GI coupled. GI means it inhibits the adenyl cyclase enzyme. Okay, and then there is no production of cyclic AMP. Okay, GABA A when acts on GABA B receptor, there is an influx of potassium in the postsynaptic neuron, whereas in presynaptic neuron, there is inhibition of neurotransmitter release. Okay, so we'll see what happens when it acts on GI coupled receptor present on postsynaptic. So here you can see. So this is a GI. So whenever a neurotransmitter acts on this GI coupled receptor, what happens? There is a inhibition of this adenyl cyclase enzyme and there is no production of cyclic AMP. And also it opens potassium channel and inhibits the neuronal calcium channels. Okay, it opens the potassium channels. Then what happens? It causes the hyperpolarization and there is a which results in impulse generation. Okay. So that's it. Here we can see there is a opening of potassium channels and there is an increase of potassium influx and there is a depression in impulse generation. Okay. So now we'll see the structure of the GABA A receptor. Okay, this is a GABA A receptor. So it has two beta subunits, two alpha subunits, and one gamma subunit. See, this is a gamma, this is alpha, beta. Okay, this middle one is the ion channel through which the chloride ions pass. Okay. First, we'll see the positions of these different sites on GABA A receptor. Okay. The position between the alpha and beta is called as GABA binding site. Okay, GABA binding site location is between alpha and beta. Whereas benzodiazepam binding site is in between alpha and gamma. Okay, benzodiazepam binding site is between alpha and gamma, whereas barbiturates binding sites are on alpha and beta subunits. Okay, on alpha, the pockets of alpha and beta subunits. Just remember this location of this binding sites. Okay, GABA is in between alpha and beta, and benzodiazepam is in between alpha and gamma, whereas barbiturates is on alpha and beta. Okay, and there is a modulatory site also. Okay, in glutamate there is a modulatory site where a glycine binds. Okay, here also there is a modulatory site. Now we'll see the functions. What happens? The responses. Okay. Barbiturates when bind, what happens? It prolongs the duration of opening of chloride channels. Okay, it prolongs the duration. Whereas in benzodiazepam, what it does? It increases the frequency of opening of chloride channels. Okay, it just increases the opening of chloride channel, whereas barbiturates it increases the duration of opening. Okay. Just remember these 2 points. Because barbiturates binding site is on directly alpha or directly on alpha and beta where the GABA binds. So there will be an increase in duration. Whereas this benzodiazepam, this site is in a different place, at a different place. So it increases the number of times of opening of chloride channels. And on modulatory sites, whenever on modulatory sites, propofol, alcohol, like these kind of substances bind to this modulatory site. So when they bind it and they enhance the activity of GABA. Okay. So whenever GABA binds, what happens? There is a hyperpolarization. See, there is an influx of chloride ions and it causes hyperpolarization of that postsynaptic membrane. And there is an inhibition. There is a depression. Okay. Next, we go to the receptor and the agonist and antagonist acting on them. Okay, first GABA receptor. It has three binding sites: GABA binding site, benzodiazepine binding site, ion channel, and also modulatory site. Okay, on modulatory site we have alcohol, propofol, these drugs. Okay, and GABA binding site, the agonist are misonol and gabaxadol, whereas antagonist bicurculin and gabazine. And on benzodiazepine, we have benzodiazepam and flumanazole. Okay. And on ion channel, we have antagonist as picrotoxin. Okay. This inhibits the opening of the chloride channels. Okay. See, if we don't want the inhibition, if we want the excitation, then we can give these drugs. These drugs blocks the chloride channel, so there is no influx of chloride ions, there is no hyperpolarization, and there will be no depression. Okay, like that. And here we can see GABA B receptor. GABA B receptor, the agonist is baclofen and the antagonist is baclofen. So now we'll study about the next neurotransmitter, dopamine, which is an excitatory neurotransmitter. Okay. So here the dopamine receptors. There are 5 kinds of dopamine receptors. Okay, they are G protein coupled. Here D1, D5 are GS coupled, which stimulate the adenyl cyclase, and then there is an increase in production of cyclic AMP, and the final response SS stimulate. Okay, and then GI D2, D3, and D4 inhibitory. I for inhibitory. They inhibit the adenyl cyclase enzyme, and there is a no production of cyclic AMP, and it results in the cellular effects based on the location. Now we'll say the location of dopamine producing neurons. Okay, this dopaminergic pathways are of 4 types: nigrostriatal, mesolimbic, mesocortical, and tubero-infundibular. So in this nigrostriatal pathway, out of these 4, nigrostriatal pathway is the major pathway which produces the dopamine. Okay. Now, in nigrostriatal pathway, the dopamine producing neurons, they extend from substantia nigra to dorsal striatum. It means the cell bodies from the substantia nigra, they extend to the dorsal striatum. Okay, they end with their axons extending till this dorsal striatum. Okay, and this pathway is mainly responsible for motor control. It is nothing but a movement. Okay, if there is any damage to this pathway, it results in rigidity, tremors, and also Parkinson's. Okay, Parkinson's disease is due to the decrease in dopamine levels. Okay, next, the mesolimbic pathway. Here, the neurons, the cell body of neurons start from this ventral tegmental area and they end with their axons in a ventral striate. Okay. And then here, mesocortical pathway also, they start from ventral tegmental area and end in ethical part of the cortex. And these 2 pathways are responsible for behavior control. It is nothing but motivation, emotion, thinking, cognition, nothing but memory. Okay. So here, if there is an increase in dopamine levels in this mesolimbic pathway, then it leads to psychosis. Okay. And then the 4th one is tubero-infundibular. Here, the neurons start from the hypothalamus and then with a median eminence and pituitary. And this pathway is mainly responsible for hormonal control. Okay. And dopamine, for example, dopamine inhibits the secretion of prolactin from anterior pituitary. Okay. Here, dopamine inhibits the secretion of prolactin from the anterior pituitary. And the next one is the next neurotransmitter, which is serotonin. It is also an excitatory neurotransmitter. See, only two are inhibitory. What are those? GABA and glycine. Except those two, all are excitatory. So first we will see the location. Here the location is RAFin nucleus and the receptors all are GPCR, G protein coupled except 5HT3, which is an ionotropic. Okay. And then these are of 7 types. Okay. The first type is inhibitory. The first and that's it. The first one is inhibitory, whereas 2, 3, 4 are excitatory. Okay. So here the 5HT1 is inhibitory and they are located in RAFin nuclei. And then 5HT2, they are excitatory, located in cortex and limbic system. And 5HT3, excitatory, area of proxima of brain. And 5HT4 is also excitatory, gastrointestinal, present mainly in the gastrointestinal tract. So these are the final responses. Okay, if 5HT1, if any ligand acts on this 5HT1, there is a decrease in serotonin release, whereas on 5HT2 it leads to hallucinations. On 5HT3 it causes vomiting. On 5HT4 it causes increase in gastrointestinal motility. Okay, like that. And 6, 7 are present in cortex, limbic system, thalamus, hypothalamus, and 5HT6 is mainly responsible for cognition, whereas 5HT7 locomotion, sleep, etc. Okay. Here we will see the drugs, 5HT agonist. Okay. A decrease in serotonin causes depression. Here the 5HT uptake blockers are fluoxetine and 5HT releasers are fenfluramine. Okay, the drugs which causes the release of 5HT are fenfluramine and the reuptake which inhibit the reuptake are fluoxetine. Just remember the drug names. So 5HT agonist 1, 2, 3, 4. 5HT1 agonist are buspirone and somatroptan. 5HT2 LSD and alpha methyl hydroxytryptamine. And 5HT3 2 methyl 5-hydroxytryptamine. 5HT4 cisapride, enzapride and metoclopramide. Okay, you just revise these drugs classifications every day so that you will not forget. That is the only way to remember all the drugs. Okay. And the next one is 5HT antagonist. So here the prevention of synthesis is done by blocking the tryptophan hydroxylase. Okay. Because in a synthesis process, in serotonin synthesis process, we can see this is the enzyme responsible for the synthesis of serotonin. So if we prevent this enzyme, we can stop the synthesis. So the enzymes which inhibit this enzyme are the drugs. The drugs which inhibit this enzyme are parachloropheniramine and parachloroamphetamine. And then the depletion of storage site is caused by receptor. It means it prevents the storage of this neurotransmitter in the synaptic. That's called depletion of storage site. Okay. Here the 5HT antagonist. 5HT1 antagonist is pipirone and 5HT2 is methysergide, cyproheptadine, ketanserin. And 5HT3 are ondansetron, granisetron, tropisetron, and all these are also act as antiemetics. Because previously we have seen on 5HT3 receptor, if any ligand acts, it causes vomiting. The final response is vomiting. So to prevent the vomiting, we can use this antagonist, 5HT3 antagonist. Okay. And on 5HT4, we have tropisetron. So this is the final conclusion of all the neurotransmitters. Till now we have studied. So here I have given some shortcuts. So here D means direct, ID means indirect. Means direct neurotransmitter is dopamine and indirect neurotransmitter is phenylalanine or tyrosine. And here VMA is vanillyl mandelic acid and DHMA is dihydroxy mandelic acid. And here in the dopamine, if the dopamine is metabolized through the enzyme MEAO, monoamino oxidase, then the final metabolite is deoxyphenyl acetic acid, okay, DPAA. And then if it is metabolized by COMT, carboxy-O-methyl transferase, then the final product is homovanillic acid. Okay. And now if we come down here, we have histamine. Histamine, if it is metabolized through N-methyl transferase, the final metabolite is methyl imidazole acetic acid. And if it is metabolized through diamino oxidase enzyme, then the final metabolite is imidazole acetic acid. So this slide is very important. The location of the neurotransmitters and their starting precursor and the final metabolite. Okay. And this is also very important one. I think you have understood. So here we can see different types of receptor and to which G protein it is coupled. So this is a very important slide. So now from yesterday's videos and from today video, what we have to focus on more. The first thing is focus on last two slides. The location, starting precursor and final metabolite, and the next slide. And then focus on the drugs, the agonist, antagonist acting on different receptors. Okay, these points are very important. Okay, from this they ask from these points only. Okay, and also which neurotransmitter is excitatory and which is inhibitory. Okay, that's it. If you focus on this, it's enough from these topics.